The protein, unlike many other proteins that have been targeted for cancer therapy and other therapies, doesn't have any obvious pockets where a drug can bind. So it's been described as a squishy tennis ball. And so it doesn't have a sort of a hole where a drug can fit in and block its function. So finding a way of getting small molecule drugs to actually interact with a squishy tennis ball is just technically very difficult because the methods just weren't around until relatively recently. And then people had found compounds previously to that which had bound, but without a detailed knowledge of the protein, they didn't really go anywhere. And then things picked up in 2012, actually, when a group at Genentech and another group found fragments of chemicals that could bind to the RAS protein using new technology, which is very sophisticated. And then Kevan Shokat at UCSF found a way of attacking one particular form of the KRAS protein through an amino acid called cysteine, which is exposed in the protein. And that cysteine could be attacked with a chemical that reacts directly and covalently with the cysteine. So that gave a handle on a drug that bound to the protein for the first time. And then from that and other parallel approaches, it sort of the whole thing just crumbled, and more people found ways of getting into the protein and more versions of it. And so over the time since the 2012, 2013 timeframe, there's now 30 or 40 different drugs that bind to KRAS in slightly different ways, but all in using the same principle.